So class, let's continue our discussion on antibiotics and antimicrobial drugs. All antimicrobial drugs are typically going to target some part of the microbe that's not present in the eukaryote, or the host that's being infected. So if we look at this antimicrobial drugs, sometimes they try to disrupt the cell processes or structures of the bacteria. Sometimes they try to inhibit viral replication. Sometimes they try to interfere with the function of the enzymes required to assemble these or synthesize the macromolecules. So if we look at this first method, or this first target up here, cell processes or structures of the bacteria, fungi, and protozoa, these different organisms, since they are not eukaryotes in the bacterial cases, and these guys, the fungi and protozoa, have very different cellular structures and processes compared to our typical animal cells. There are going to be a lot of metabolic pathways or targets that we could selectively deactivate with hopefully minimal side effects or side reactions in the eukaryotic cell, because we're ultimately focusing on having a human not being infected. Inhibiting virus replication is typically going to focus on targeting focusing on targeting ribosomes, or we could have the cells not be able to fuse with the virus. If we look at interfering with the functions of enzymes for synthesizing or assembling macromolecules, there are different flavors of the same enzyme. For example, lactase dehydrogenase is an enzyme that's present in many different organisms. But depending on the organism, lactase dehydrogenase is going to have a slightly different shape and slightly different conformational structure to its active site. So we can design a drug that will selectively bind to one kind of lactase dehydrogenase compared to another kind of lactase dehydrogenase. So we can target one kind of organism and not the other kind of organism. We can destroy structures already formed in the cell, particularly when it comes to targeting the cell wall. There are many antimicrobial drugs that will either target a peptidoglycan cell wall or a chitin cell wall formation, and since we as animals do not have either peptidoglycan or chitin or any cell walls period in our cells, that makes a very easy target for antimicrobial treatment. A main goal of our antimicrobial drugs is that they need to be selectively toxic. We want to kill the bad guy but not hurt the good guy, or minimize the amount of side effects or side reactions that take place. So we don't want the medication to inhibit any metabolic processes for us in our eukaryotic cells. If we look at drugs that have excellent toxicity, they're going to block the synthesis of a cell wall, particularly a bacterial cell wall. These drugs are incredibly toxic to bacteria, but have very, very little side effects to human beings. Since we don't have peptidoglycan in our cell walls, generally speaking, the penicillin class of drugs has very little effect on us. Drugs that do have effect on us, though, are going to act on a structure that's common to both us and the bacterium. So I mentioned how, for example, when we are targeting a virus, sometimes with an antiviral drug, we're going to try to inhibit the the ribosomal activity, so we can't synthesize viral proteins. And that works really nicely for inhibiting viral reproduction. The problem is we are inhibiting our own ribosomes, and we need our own ribosomes to make proteins for us. So that's kind of a darned if I do, darned if I don't approach for antimicrobial treatment. So if a drug has a characteristic of the infectious agent, and the infectious agent is very similar to its host cell, the selective toxicity becomes very, very difficult to achieve. We don't spend a lot of time talking about this, because it's not necessarily a microbe that attacks us, but cancerous tumors are an excellent example. One of the reasons why chemotherapy is such a big deal for people and has so many terrible side effects is because we're trying to kill human cells but not kill other human cells at the same time. We're trying to kill the human cells that are in the cancer or in the tumor, but not kill the human cells that are forming appropriately. And in those situations, it's very difficult to design a drug that targets one human cell type and not targets the other human cell type. That's the extreme end of the spectrum in terms of very toxic drugs for human beings. Thankfully, viral, bacterial, protozoal, and fungal cells are going to have enough differences that we typically don't have that kind of toxicity. So if we look at chemotherapy, and in this sense, when I say chemotherapy, I use a chemical to treat a sickness. I do not mean cancer medicine. So when I say chemotherapy, I mean just any kind of antimicrobial. The goal of a chemotherapeutic drug is to identify the structural metabolic needs of the living cell. So we're trying to identify the needs of the cell, remove those needs, or disrupt or interfere, make those needs not be met, so that that parasitic cell is no longer able to cause problems for us. Antimicrobial drug categories are going to focus on cell wall synthesis. I think I've said that 3 or 4 times already this presentation. We also focus on nucleic acid structure and function, the inhibited of protein synthesis. If we look at bacterial and archaeal ribosomes, they have different structures than eukaryotic ribosomes, so that makes them an easier target. We want to interfere with cell membrane structure and function. And then we can also focus on the inhibition of folic acid synthesis. We as human beings, if we're fully grown and no longer developing or increasing in mass, so let's say you're an adult, you're done growing, folic acid is not very essential for us. But for microbes that are rapidly growing and reproducing, folic acid is pretty essential. So here is a summary table focusing on all the common targets for antimicrobial treatment. We can see that we can target the prokaryotic ribosome. We can either target the large subunit or the small subunit, and we can see here are the drugs that do that. So erythromycin, cinaricid, pleuromutilins are all going to target the large subunit. The small subunit can be targeted by tetracycline, our aminoglycosides, or our glycocyclines. And then some drugs that will affect both the largest subunits include the linezolid. Some drugs are going to interfere with transcription translation, such as the quinones, the rifampin. Some drugs are going to focus on the cell membrane, such as the polymyxin and the daptomyxin. And then, you can imagine, our cell wall inhibitors are going to be the most common one, because the cell wall is a very logical target for an antimicrobial drug. So we have our penicillins, our cephalosporins, our carbapenems, our vancomycins, our bactricins, our fosomycins, and our isoniacids. All are going to focus on cell wall production and inhibiting this production of the cell wall. And I'm pretty sure I mispronounced half of those antibiotics. So here we can have a summary table. So here we have a nice figure, and then I took the information from this figure, and your textbook put it in this table. What do I want you to know from this table? I want you to know that drugs that target the cell wall include penicillins, cephalosporins. Over here, the uses and the characteristics and the specific subgroups within those drug categories, I am not going to expect you to know for the exam. I want you to focus on that. Penicillin targets the cell wall. Cephalosporin targets the cell wall. And our carbapenems also target the cell wall. And then these guys over here, the bactricins, the isoniacids, the vancomycins, and the fosomycins also target the cell wall. So know what targets the cell wall. Our aminoglycosides target protein synthesis. And then our tetracyclines, glycocyclines, our macroglides, and a few other miscellaneous drugs all target protein synthesis. Folic acid synthesis focused on our sulfonamides. Our sulfonamides are an old category of antimicrobial treatment. I believe that these predate penicillin, but don't quote me on that. Our fluoroquinones are also going to target DNA synthesis. Our polymyxins are going to target the cell membranes. So, concept check class. Which of the antibiotic modes of action will have a least toxic effect on a human cell? So think, what is present in a bacterial, prokaryotic cell, or in what is present in a eukaryotic cell? Do human cells, let's back up here. We have antibiotic A, acts on DNA replication, antibiotic B, acts on the cell membrane, or antibiotic C, acts on the peptidoglycan cell wall. So what structure here is present in bacterial cells, but not in human cells? Do human cells have DNA? Hopefully you say yes. Do human cells have a cell membrane? Hopefully you say yes. Do human cells have a peptidoglycan cell wall? Hopefully you say no. Now, if that hint wasn't big enough for you, you should pause the video, rewind it, find the answers in your notes, in your PowerPoints, and then confirm your answer. 1, 2, 3, 4, 5. The correct answer is C. An antibiotic that acts on the cell wall is going to be the least toxic on the human cell.